Broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You have logged on to the Nelson Labs webinar, ISO 10993-1, key update on the new revision of this critical standard. Now, we just have a few housekeeping items that we'd like to take care of before we start. If you missed one of our webinars or would like to refer back to one, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can also receive notifications for upcoming live and on-demand webinars by following Nelson Labs on social media, Facebook, and on LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will also find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 900 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve your patient outcomes and minimize risk. So let's get started today. We are joined by Thor Rollins and Nicholas Cristiano. Thor is a certified microbiologist and specializes in the selection and conduct of in vitro and in vivo biocompatibility tests. He actively speaks on biocompatibility related topics through Nelson Labs external seminars, webinars, and trade shows. Thor presents on many biocompatibility topics at industry conferences and events and has published many articles on these topics. He is a participating member on All Association for Advancement of Medical Instrumentation, AAMI, TC 194 and 10993 ISO committees, and plays an active role along with the FDA and regulatory committees developing standards, discussing biocompatibility methods, and voting on changes to those standards. As one of a select group of experts in the industry, Thor's participation on the committees offers an insight on industry changes and helps prepare clients for changes in testing. Nicholas serves at the global market segment lead for biological safety at Nelson Labs. He is responsible for driving strategic growth, leadership, and market direction across biocompatibility and extractable and leachable testing segments. Nicholas has over 12 years of experience in the medical device biocompatibility field, and prior to Nelson Labs, he held scientific roles within biocompatibility segment at a major orthopedic medical device manufacturer and within pharmaceutical drug development in the oncology space. Nick is a part of the standards development with AAMI and participates in many working groups, as well as supporting the development and round-robin efforts towards multiple in vitro alternatives. We do encourage all of your questions. Please submit them at any time in the questions box, and Nicholas, and Thor will try to answer as many as they possibly can in the last 10 to 15 minutes of this webinar. Shortly, we will have a poll question regarding genotoxicity evaluations. We ask, it's a simple question. We ask that you take a moment to answer that one question when it comes up. So now, beginning today's presentation, I'd like to turn the time over to Thor and Nicholas.
Thanks, Mike, and thank you everyone for attending the webinar today. Nick and I are super excited to talk. Well, we're always excited to talk about biocompatibility, but specifically when we're talking about this new 10993 standard and its evolution, it's something that we're always excited to kind of address and kind of keep you guys up to date on what's happening to the standard and what we see as the future impact to our customers. I just want to make another quick plea for what Mike said is we'd love to hear your questions, and even if Nick and I can't have time to get to them, we promise that we'll email back answers as best we can to try to answer any questions that come through the Q&A. So let's go ahead and jump in. Let's get started. So this is a quick timeline about how this standard has evolved. So as you can see, the standard that we currently use, the 10993 2018-1 2018 version, was published in 2018, and it's the current standard that we use. Shortly after that standard was issued, there was a new project registered with ISO to basically start rewriting the standard, and that was the 21st of August 2021. And that was a kind of a quick turnaround time. We had just finished 2018 publication, and then pretty quickly afterwards there was a new project, and that project drastically changed the 10993 as we know it, and we'll go through those. So then we had a working draft initiated. That's when the first time that the members of the ISO committee draft for -1 or committee started to see the new draft and look at the changes. And then I went through mini ballots, which are voting, comments, things like that. So you can see the dates where we got either comments due or comments submitted. But I think the thing that matters the most is the last two boxes up there. So in March of 2024, we met in Japan, and we worked a whole week every day as an ISO experts to try to get this standard to a point where we could get global acceptance. And at that point, it moved to a draft international standard. So once again, it was released to the the governing bodies of the different countries, and had different comments done on that disc. And then in October of last year, we met again in Paris for a week, and then we went through all those comments. We argued and discussed about the things that we're going to talk about, but then probably the key point there, it was voted to be moved to an FDIS, which stands for Final Draft International Standard. And why that's important is because at this point, an FDIS cannot have any more technical changes.
And as Thor mentioned, what's really interesting here, and I think something to really focus on as we move forward and talk about the rest of the standard and these changes, is that if you see that bracket down there, it shows the average about 1.5 years, right? So from the time that the committee draft was first balloted, so back in July of 2023, up until when Thor had just mentioned the draft had moved into FDIS, you're talking about a 1.5-year window there. So this is a very, very fast speed for such a large and in-depth standard as the one we're about to review.
Yeah, I don't know if anybody on the phone call has ever worked in the ISO committees before, but the fact that this is such a drastic change to the current standard, and we got it, and we're out at FDIS in a year and a half is pretty amazing. I've never been part of something that fast. So Nick is totally correct. So what we think would be the biggest, so we're going to go through the changes and try to do detail where we can. We don't have time to get into detail on every single point that's changing, but we will spend the majority of the time discussing these 4 main points. These are the points that we believe will have the biggest impact on our customers. And what this is additional endpoints to be evaluated. So that includes genotoxicity, new guidance about cumulative contact duration, which I think we'll have a lot of questions about. So we'll see how that goes. Another thing that kind of scares me a little bit about how it's going to be enforced is foreseeable misuse. And then the last thing we're going to get into some detail is end-of-life evaluation. There are other things we'll talk about that are going to be different in the standard, but these 4 things Nick and I think will have the biggest potential impact to our customers. So these are the ones we're going to spend the most time, get the most in depth with. So one question I always get asked when we go through this is we have all these voting points within the standards life cycle. So how did the US vote? Well, this is actually the ballot from the very first committee ballot. And as you can see, Germany and US has voted no. Well, the reason I bring this up is because the US has pretty much voted no every single time on this when it's been balloted, including the last time at the FDISC. And when we go through this, we'll kind of point out what I believe, and I am one of the members who are voting for this. But I will kind of mention why my thoughts, or why I voted no, and why I think the U.S. voted no.
Yeah, and holistically speaking, the U.S. group doesn't feel like this standard may be completely ready for revision in its entirety, just because, as we briefly mentioned, the length of the standard, right, and the in-depth and the complexity of the standard, again, as we covered, really was updated in 1.5 years, right? So did we have enough time to really, truly dive deep into deciding on which direction the standard should go? And as a byproduct of the U.S. voting no, we do expect that the FDA may issue a guidance document on this new revision as well.
Yeah, either FDA or AMI or something. There'll be additional. So to Nick's point, I don't think anybody believes that this standard is complete yet, but there's some philosophy that we should release it and then continue to work on correcting or including more guidance into the standard. The U.S., especially me, we feel like we should it's not like the 2018 version is broken, there's a lot of great concepts in this FDISC that we should work on and evolve and give more guidance on. So it's just a kind of a different view on when we should release something, either a working draft kind of, or something that's more complete, like Nick said. So that's kind of in a heartbeat why the vote no happened each time. So let's dive into it. Nick and I, we're going to cover what's in this potential 2025 FDISC that's going to be hopefully released any day now. So we've got lots of clues that it's going to be coming out soon. So we're just waiting for that to be officially released. So what I tried to do here is I tried to highlight different categories where I thought there was large changes, or potentially not even large, some small changes, just so that when you guys get the FDISC copy, you can go through and read these particular ones. If you love standards so much that you want to read the whole thing word for word, please do so. I'm never going to take that pleasure away from people. But if you're one of those people who just do this for your job, these are areas you might want to focus on. Okay, so there are some changes to scope, and particularly there's been some changes, some differences in concepts. One that we're going to spend a little bit of time on is highlighted in red, which is evaluating significant changes through the life cycle that could impact your biocompatibility. This is something that we've seen recently from notified bodies, where companies have come to us and asked, or saying, hey, our notified body is asking us to justify how our biocompatibility has not changed through the life cycle of that product. And that could be shelf life. It could be if it's reprocessed, and we'll get into that a little bit later too. But this now is being added to the standard. So this concept is now something that we have to evaluate. I would say, for me, like I said, we're seeing this already being asked by especially notified bodies. And there's different ways to do this. In fact, last week at the Society of Toxicology, some individuals from Teleflex did a great job at presenting how they evaluate the impact of the life cycle of a shelf life of their products, and it had to do with some different chemistry tests. So there are companies who are already looking how to do this. If you have specific needs on this, obviously you can reach out to people who are doing this right now, including us. We can get you in contact with people or help you through this, but that this is something that you're going to have to evaluate. And I do want to kind of emphasize here, it is going to be device dependent. So I think everybody would agree that a piece of like titanium sitting on a shelf for 5 years is not necessarily going to change its biocompatibility. If anything, you have surface residuals that might be volatile or semi-volatile that could be actually off-gassing over time, and maybe you're even getting better over that shelf life. So, but if you have a polymer that has different additives into it, like stabilizers or other types of additives to this polymer, now that shelf life, the leachables from that device would probably change over the shelf life of that material. I'm not saying it's going to get bad. I mean, that's the point. You're not going to potentially have toxic material being generated, but we're not having those answers today either. So there has to be some evaluation being done, either written or some testing, to be able to show that your materials on a shelf life are not changing over time.
Yeah, and a good point to that too, Thor, is also the standard does, this evolution of the standard does also allow for us to leverage some clinical data, so some post-market surveillance data that may be existing for these components or materials. Not something I think historically we have focused so much on in the past, because biocompatibility has been so focused on that time zero date, right? And that's where we've established our clinical relevance and clinical safety. And so now being able to kind of dive into post-market surveillance data to help us support these written evaluations in lieu of potential testing to come, or maybe also in partnership with testing as well on what the shelf life of those more so biodegradable materials or polymeric materials, as you mentioned, Thor, makes sense. So it is really nice that we will be able to start diving into what post-market surveillance may actually help our evaluations in the future as well.
And then one more point on this slide, and then we'll move on to the next one. It's not highlighted in red, but it's just something that I think some medical device manufacturers out there might think about, which is we are kind of including more about who contacts these devices. We added clinicians in 2018 to the standard because we're worried not just about the patients, but the people who could contact these materials and use them every day. Well, now devices are going out of the hospitals, and more of them are going into the home. And so our contact is not just patients or even nurses or doctors. It could be parents, it could be siblings, it could be spouses. So these are things now that we have to think about at the home. They're not trained, they're not necessarily educated in how to use these medical devices. So these biocompatibility risks could be a little bit different for some of these devices.
And safe to say, we're kind of shifting the thought process from just patients to kind of consider all users, right?
Exactly. Okay, so these two things here, this slide might be up for a couple minutes because we're going to be talking a little bit about foreseeable misuse and then genotoxicity. And this is where we're going to have a poll in a minute, and I'll talk about the poll when we get to genotox. But first, we're going to talk about foreseeable misuse. And this is something that was a pretty key discussion in the ISO committee. There's a lot of medical device manufacturers who rightly so are pretty worried about what foreseeable misuse might mean for biocompatibility. So this is directly out of 14971, which we'll actually talk about the merger between 10993 and 14971 in a minute. But many companies have to think about foreseeable misuse when it comes to their medical device when they're looking at the 14971 thought process. But for biocompatibility, this comes into line when you start thinking about how long a medical device could be used for, but could you see it being used longer than intended? Or something we see all the time is patient populations. We say, hey, just use on adults, but that they could be used on a more sensitive population. And so now do you have to look at the biocompatibility risks of that population? And I'm sure nobody on this call has done this because you guys are all great medical device manufacturers, but I have seen a couple people go, you know what, we don't want to have our device be long-term contact, so we're going to put in our IFU do not use more than 28 days, but they know that doctors are using it more than 28 days. So those kind of things are why the foreseeable misuse has been put in. I think the fear it's kind of Pandora's box. There are certain applications I think everybody could agree of possible foreseeable misuse, but now are you going to have a regulatory body kind of come up with a foreseeable misuse that you did not anticipate? And is that now going to make your testing longer, have to go back and repeat stuff? I think that's the fear for medical device manufacturers about that.
Yeah, and in addition, Thor, I would add, I think a lot of people on this call, maybe don't have a lot of experience with understanding misuse and device failure because in a lot of cases, maybe company dependent, manufacturer dependent, but these responsibilities may fall within the engineering departments, right? More upstream. And so what used to be maybe more so considered within design verification and validation as in misuse and failures, right? And identifying those potential failures now also has a little bit of overlap with our biocompatibility teams now here at the manufacturer as well. So I will say, I don't know if I want to call it an unpopular opinion, but I like this addition. I think it makes sense, right? We should be considering these things. We should be considering all aspects of how the end user could be contacted, right? And what that indication could turn into or could be, interpreted as. However, I'm not sure how well this is going to work with understanding what will be expected from, as you mentioned, how much is maybe over challenging ourselves here and understanding misuse and potential device failure.
Yeah, I agree, Nick. Actually, as a patient, I like the idea of the fact that if surgeons or clinicians are using certain products off-label and that's routine, I would hope that they are assessed for that use as a patient. So I do agree with you there. I think there's just as a double-edged sword here as far as being able to hopefully understand what foreseeable misuse really means in these situations. The next one is probably another big impact to our customers, well, certain customers, which is there are more devices that are going to be requiring to have genotoxicity assessments. So, and we'll get into this more as far as actual the new requirements for device, but in general, if you have a risk for systemic toxicity, then you must address genotoxicity. So this starts to impact devices that have any type of contact with circulating blood, regardless of duration. It also, if you start looking at like mucosal membrane or tissue contacts, there were some contacts in there like prolonged contact in certain applications that did not require genotoxicity testing that now will. So the way you would look at this is if you've had to do acute systemic toxicity or subacute subchronic toxicity in the past for your device or assess that endpoint, you now have to also assess genotoxicity. So they go hand in hand with each other. So one of the poll we'd like to send out, and Mike's going to push it now, is if you in the past didn't have to do genotoxicity, but now you think that you do, will you think you'll have to run that test now? So the poll will be a yes/no question. So basically, if you have to do genotoxicity now or before you didn't, please push yes. If you still don't have to run genotoxicity, push no. So that's the poll that we're looking to push out. But the concept here, and this actually goes to our next page a little bit. Oh, and I lied. It's here soon. So we'll just go. We talk about it later on. But the concept here is that if you go systemic, then that is a potential to mutate your genes and cause long-term issues from a genotoxicity potential. So if you have a systemic toxicity risk, in theory, you have a genotoxicity risk. They should be hand in hand with each other. And so that's why this standard was manipulated that way. Okay, so one of the concepts we added to the standard that is actually something I'm really happy about, and it's something Nelson has done in our risk assessments for years, and I think a lot of people have done, but it's the concept of a medical device family. So this has to do with using your biological evaluations, your BEPs, or your maybe a biological risk assessment, which is the concept. If you remember, the heart of biocompatibility is you look at the materials, you look at possible residuals from the processes, and how the device is being intended to be used. So those three points are added together as your evaluation. Well, if you have devices that have very similar, if not exact materials, very similar, if not exact processes, you can actually start to put together a family and look at a worst case device to test for biocompatibility that represents that family. And like I said, this is something that we've done before at Nelson. I'm sure people have before too, but now we've actually added those concepts to the standard.
Yeah, and a huge benefit, Thor, too, for companies, manufacturers that have a lot of SKUs, right? So we have a ton of devices within certain internal families, perhaps, of the manufacturer, with very little changes or, the same manufacturing processes, the same downstream processes that allow us to conduct a more master evaluation, if you will, similar to how some of the call may have familiarity with in the sterilization world as well.
And actually, that's something I want to say, because obviously the sterilization group has done family groupings for years, and that concept's the same. But just so you know, what makes a sterilization family grouping is not necessarily the same thing that makes a medical device biocompatibility family grouping. So you might have to have multiple family groupings, one for sterilization, because that's the one you're looking at, kill and the materials and things like that. But biocomp is a much different consideration. So same concept, but just different inputs into that consideration. But the standard walks through that for you. But good point, Nick. Okay, this is one that I think could have an impact in the industry. I'm hoping that there's some evaluation for risk here. We'll talk about it, but there was a need or a want from some of the ISO experts to put into the standard more background needed, more expertise needed for people who are performing biological evaluation plans or biological risk assessments or biological evaluation reports. The purpose of this was that some notified bodies or regulatory bodies, I should say, were mentioning that they were seeing some pretty poorly written assessments. And so they wanted to put in the standard more kind of emphasis on who can write these documents. That became kind of an interesting discussion because there's not really someone you can say, like, for example, if I'm looking for someone to do a tox assessment, I would say you need to be a board certified toxicologist. And everyone kind of knows what that means, and there's a good indication of what that means, and so that can be something understood. For biocompatibility, that's a really broad kind of skill set. And so what we have in the standard is that it should be planned, conducted, and reported upon by a knowledgeable and experienced professional, appropriately qualified by education, training, skill, and experience, capable of making informed decisions based on scientific data and a knowledge of medical devices. And this is the important part. Qualification of evaluators shall be documented. So when you write your BEPs or your BERs, you have to submit a CV now to be able to justify the person who's signing off on that is qualified to write that document. And so that is something that I think, hopefully there's enough qualifications out there for people to be able to still do this. I just hope it doesn't limit the amount of, make a squeeze on the capacity of people to be able to write these documents.
And I think this is something that we've been seeing coming, down the path for quite some time for part one of these changes, these isolated changes. Long gone are the days of perhaps a regulatory affairs specialist just providing, the checkbox approach to completing and conducting biocompatibility testing, right? Understanding, using the tables, the historical tables of clinical indication, exposure, type, and time, and then checking the boxes, ordering the tests, right? And kind of adding the tests to the annex to the 510K. So I think we knew that this direction was coming. I will definitely second your thoughts on that, Thor, of, what will be enough, right? We could see on this slide here, we threw out a few titles, just, a quality engineer, right? A toxicologist, a particularly certified toxicologist, right? A subject matter expert, and then maybe even how do we define whether you are a subject matter expert. So as Thor mentioned too, I think you've seen a lot of submissions recently require CVs, and we're hearing more and more response that is required as a appendix, if you will, to the 510K or the biocomp submission. And so again, something that we've seen coming, I think more to come to understand what that qualification looks like. And this may be something that just ends up being a little bit reviewer dependent, which we all get a little heartburn over, but I think there's a reality there. Yeah,
amen to that. So one of the biggest reasons the standards came out is the desire to merge 10993 with 14971. And in 14971, most of you who've used this standard before, there's a need to look at risk through risk analysis, risk evaluation, risk control, risk mitigation, things like that. And so the concept is let's take these risk evaluations and apply them to biocompatibility. And that's easy to say. In fact, you might even think that sounds like a great idea. The hard part about that is how, right? So like the risk estimation part especially is like really difficult when you actually get to the doing it. So you might think, sure, I could estimate my risk. That's why I perform biocompatibility tests to look at sensitization or systemic toxicity. So that's my risk estimation. But what does that mean from a practical standpoint? We we tried in the standard to do that. So we actually worked on an annex where we tried to give examples of how you could look at probability of risks and the severity of that risk, and then also do some kind of scoring so that maybe you could look at your device through a numerical value. But when we worked on the comments and going through that as an ISO committee, it was no one could agree on things. And because of the timeline they were trying to get into, they actually just removed the annex. And that's actually one of the main reasons I, in the US, voted no, as to Nick's point earlier, is we just don't think it's ready yet. We think there should be more work done, especially on this risk estimation part, to be able to help the users of this document understand what it means to do risk estimation from biocompatibility.
Yeah, absolutely. And to Thor's point, this is why a year and a half, as crazy as it sounds, isn't quite enough time for the committees to appropriately identify how to do this. Again, it's a standard, but how to do this as objectively as possible, right? So we all know that there's some subjectivity within the standard, right? We have to read between the lines occasionally. We have to understand how regulatory authorities or the FDA or others around the globe who do honor the standard, how they interpret the standard as well, for those who do recognize it. However, one thing that's important that's missing here too, right, is residual risk, right, and understanding that. So those who are familiar with risk assessment practice in general, right, we have to identify the risk, right, we have to figure out how to mitigate the risk, and then we should objectively understand how to determine what the residual risk is left. And maybe even from a toxicological perspective and as an entirety, a biological safety perspective, what is acceptable, right? What limit of residual risk would then be acceptable? So again, a year and a half, just not quite enough time, right, Thor?
And it wasn't because people, there are individuals that worked very hard to try to come up with examples of risk estimation. But it's just, yeah, we need more time to come up with practical examples to help with this. So the standard is right now not really complete, in my opinion, on giving people guidance on how to do risk analysis and risk estimation per 14971 for biocompatibility. Okay, just for time, I'm going to skip over this a little quick. There is some things about biological evaluation plans. And what needs to be in there. There's a new flow chart that we added on how to write a BEP. So for those of you that are writing biological evaluation plans, we're trying to standardize the approach a little bit more. So that was a good benefit. But I wanted to make sure that we spend some time on another part of the whole life cycle, and then also about the recategorization, which would probably be one of the two biggest impacts. So this one has to do about reusable devices. We talked about shelf life. But one of the things that's been put into this FDISC is that the validation for reprocessing should be done on the maximum number of validated processing cycles. And so the concept here, and I think like Nick said before about end of life and things like that, it's actually a really good concept. So people who make reprocessed devices like scopes and scissors and things like that, you got to remember, hospitals use these devices basically until they fall apart, until they don't function anymore, and then they throw them away. Well, our validations don't really mimic that. Our validations, they may run 6 cycles where they soil and clean, and then that's when we stop the validation for reprocessing. But what happens to the biocompatibility when that device starts to break? You have detergent buildup, materials might start to break down, things like that. And so in the standard, you now have to evaluate your biological risk over the life cycle of that reprocessed device. So what does that mean, right? Does that mean that you have to reprocess it until it breaks down and then do biocompatibility? Maybe. Or does it mean that you have to look at it from a material perspective and understand the risks from it being at the end of its life? Maybe. So the only reason I put this in here is because I don't know if we exactly know how this will be enforced.
Yeah, and I agree, Thor. I think this is a great addition to the standard, as I mentioned before. Again, how do we identify these risks is definitely, potentially Pandora's box, right? As you mentioned, detergent buildup is one of the easiest ones to think of. Corrosion, right? I think this is a question that's been asked, at least in the world of orthopedics, for a very long time in reusable devices. How much rust or corrosion is too much, right? I mean, that's just the headline in itself. Passivation layers degrade, exposing other material layers, etc.
Yeah, very good, Nick. So this one we're just going to be very high level on because of time, but you know the original drafts really went hard on replacing animal testing with things such as, and this is what the original the committee draft that was originally written had things like you should justify when you do animal tests, right? So if there is available relevant studies that have been carried out previously, or if there's existing non-clinical or clinical data, then you should not do animal testing because it would be unethical. So it came out really hard against animal testing, and to a point where he even said if there's a validated in vitro alternative, then you shouldn't even be doing the animal test. Well, this was pretty controversial. So there was even a lot of fighting, you know, with regulatory bodies and industry and things like that. So this was changed to basically reduce to say that you now just have to, we just reference part 2, which is the animal welfare part of the standard. So this part was kind of brought back a little bit, softened. So now it just talks about verbiage that you should reference part 2 when looking at animal testing. Okay, this is the one we're going to probably spend the majority of our time left on. We'll hit the rest of the stuff high level, but this is something that I think could impact a lot of our clients. And I know it has because I've talked to many of them who are not super happy about this change. So most device, or not most, a lot of devices are used repeatedly. So an IV bag is a good example. So a person goes into the hospital, they get put on an IV drip. After a time, if they run out of that fluid in the IV, a new IV bag's been put into you know, that patient. Well, if you start historically, a lot of people looked at the category of their device by adding up the contact time. So in this example right here, you had a device that contacts the patient 5 minutes a day for a week. Okay, so a lot of my customers would categorize that device by going, it's 5-minute exposure every day for a week, so 5 times 7, that's 35 minutes, so therefore it's a limited contact device. And honestly, I think the majority of my clients did that when they started looking at contact duration. Well, this standard now specifically says that if you have contact with the body, regardless of time, except for that bottom part where it says very brief contact, which is typically less than 1 minute. So if you have contact with the body for more than 1 minute, regardless of how long, it's considered a day contact. The concept there is that chemistry from the medical device is being leached into the body, and then the body's being kind of metabolizing that, and we don't know how long it's going to take for that to be, you know, rinsed out of the body. So at the next contact, you might not be 0 again, you might be additive on that chemistry. So with this now, this device that's in the example, this device would now have 5 minutes, that 5 minutes would be a day, so it'd be 7-day contact, and now it's a prolonged contacting device.
And something that you also will see that it seems to be pretty relevant throughout the standard is, you know, we haven't historically given much guidance on length of time. You know, truly within part 1, it's been potentially a sum of total contacts, right? What is that, you know, added up, right, or considered over time? But now as we've considered, you know, part 18 and extractable leachables with a little bit more pressure, you know, in the current days, we see that part 1 is also moving towards a little bit more of an understanding of the exposure time, of course, from a risk-based approach.
Yeah, and so like there's like catheters that are used for urinary aspects in the hospital that only have a couple minutes exposure, but that person could have a urinary catheter a couple times a day for their whole time there. So instead of adding up 5-10 minutes a day times 20, now that's, you know, every day is a contact time. So these are things that eventually a lot of medical devices are going to be up categorized, and we'll have to evaluate more endpoints based off this new contact duration.
And again, as you said, Thor, the concept here, generally speaking, that I like to kind of walk away from this with is that any chemistry that could be leaching out of a device, right, may have an impact on the body regardless of time.
And actually, that's the next use, which is the reason we did this is because we don't know about bioaccumulation. Now, there are discussions, right, where if you can prove that your leachables coming off your device will not accumulate over the 24-hour point, then maybe you could justify that that's not a correct categorization. I've been testing medical devices for almost 25 years. I don't know how successful that's going to be. So I think the majority of our devices, we're going to be looking at this type of categorization. So obviously, and by the way, I do want to say, we're saying evaluate. So remember, the standard does not say test. What it means is you might have more endpoints to evaluate now than you did before. I know people are screaming at me right now in the comments saying, yes, Thor, but regulatory bodies are still checkboxing. And so if I have to do this test, I have to do it. We work with this every day, right? So we know, but there are ways you can evaluate endpoints besides testing, and we have success in that. It just takes some time and effort. So I don't want you guys to think that everybody has to test all the time. It is a risk-based approach. So I just wanted to let you guys know you don't necessarily always have to test.
And again, that's also the importance of having qualified individuals reviewing these products, right? So, having qualified toxicologists really help you understand potentially if you do have compounds that could be elicited or leached from, from these materials that may bioaccumulate, right? Or there may be a subset of compounds that we know scientifically do not, right? So again, kind of reverts back to having the right qualified individuals making these decisions and helping you make these decisions on paper.
So we only have a couple minutes left before Q&A. So what I'm going to just end with is I wanted to also end with a high note. And in fact, later on down here, we have into the standard this FDA guidance, which is now NXG, which talks about skin contacting materials. So if you have those types of materials that are skin contacting, they are considered low risk, and you can just write, a justification. Well, the standard is now putting that into the FDISC, right? So now we have insignificant skin contacting medical devices. So if you are touching, if your materials that we touch every day, made out of consumer components, things like that, then it's only intact skin, then a simple written justification would be appropriate. So we're actually putting that in there. Some other things just to, and so there are new charts. We didn't want to put the actual ISO charts in there just for copyright's sake, but we did kind of mimic what they look like, where you do record more genotoxicity. That's why they're highlighted in red there, so you guys can see where there might be some increased genotoxicity requirements. And then ultimately, the last thing I want to talk about is here with genotoxicity. So as you can see, it says, shall be evaluated for prolonged or long-term contact with tissues other than blood, long-term mucosal or compromised surfaces, longer indirect contact with circulating blood, and then direct contact with circulating blood for any duration. So those are the endpoints that are going to be needed for genotoxicity. Last thing, and then we'll go to questions. We did end up reducing the need for material-mediated pyrogenicity. So if we can all cheer, that would be great. So a lot of kudos to Kelly Coleman, who put together a lot of work here, where we looked at a bunch of labs, E&L, tried to identify if we were ever seeing a material pyrogen, and be able to show that this test was basically not being ran, or was being ran for nothing. And so based on the data he presented, we were able to take out the material-mediated pyrogenicity unless you're using materials that have never been used in a medical device before. So if you guys are going somewhere in a different country and finding materials that are being made that have never been made and put in a medical device, then you still might have to run an MMP. But for most devices, this test is no longer going to be required. And then we're now going to try to use that same rationale for other tests like sensitization, which is high on my list.
Yeah, and as you said, Thor, it's really nice to see a win here in the world of MMP. Kelly Coleman's work, as Thor mentioned, actually was awarded the Best Medical Device Poster Award as well this year at Society of Toxicology. So definitely a good amount of progress there in MMP, as you mentioned, unless you have unknown materials.
Exactly. Okay, so I think, Mike, at this time, there are a couple, one last thing I'll talk about before questions. I know I said that many times, but another good thing is we've added concepts about biological equivalence. So in 1093-18, there are kind of a chart about like this, where we talk about chemical equivalence. And honestly, chemical equivalence is really difficult to get, right? Like, even if you run E&L on the same, a device from the same lot, there's going to be discrepancies on the amounts of chemicals coming off. So what is chemical equivalence? So Really what we care about, though, is biological equivalence. Does that device initiate an increased risk to the patient than the other device? So if you're looking at changes or things like that. So we put in some guidance on how to do that, how to look at the physical evidence, the contact of the device, the materials, to put together an actual biological equivalence argument, so that you're basically saying that this device does not elicit a higher risk to the patient than the original device. So that's a good addition to the standard.
Yeah, and where you would want to consider this and make sure you footnote, can I be leveraging this addition to the standard, is when we're discussing family grouping, right? That's the easiest time to probably realize that you likely may have some equivalents that you can leverage.
Yeah. Well, I wanted to thank everyone for your time. I do want to remind you, that I'm going to stop sharing, that our academy is available, and so can this webinar will be up there so that you can go in and take a look at it. But Mike, we'll turn it over to you for questions.
Great, lots of information there. Thank you, Thor and Nicholas, for your insights. There's a lot of great information there, a lot to think about. We do have a lot of questions that we'll get right into. So here is the first question. To my understanding, the U.S. had voted against progression of the ISO DIS 10993-1. Did conversations in the Paris meetings change any stance on that?
So yes, the U.S. was the only country to vote no to progress the standard to the F-DISC, which Nick and I talked about the reasons that I felt like it should be a no. So how it works is one country voting no does not give sufficient. It's not, you don't have to have a perfect acceptance. And so one country wasn't enough for the standard to not move on. So no, the standard is going to move on to an F-DISC. But like Nick said, it's going to be interesting to see what is going to happen afterwards. Does the FDA come out with additional guidance on how they may interpret the standard? And then also, does AAMI do something independent of the ISO standard? So these are all things that we're going to have to think about going forward. But right now, that does not change the F-DISC at all.
Okay, great. Here is our next question. For new product development, how would you see that testing or evaluation of end-of-shelf life biocomp can be conducted? Testing on accelerated age samples? What are your thoughts on a practice level?
Yeah, so I think it's going to be a lot like, it's going to be, to me, a lot like what we do with a lot of the aged shelf life, which we would originally do it on accelerated age samples. And then we would have to maybe look at what a real time would look like. So I think it's going to be a little bit of both. And by the way, I'm not talking about a lot of difficult tests here. So, the Teleflex I mentioned earlier, there's just some simple tests that they run to look at molecular weight differences. They look at thermal temperature differences, things like that, to try to help justify that the materials aren't changed significantly enough to cause an increased concern. But that's how I would look at going.
Yeah, and I would agree with Thor on that. I think conceptually, my mind goes to the same thought process we have with implantation studies, right? Sometimes there's a business decision that we want to submit the 510K, we want to get it under review. Perhaps we have a shorter term implant study underway, or excuse me, we have results from a short-term implant study where we may have, a 13-week or 26-week actually underway, right? So showing the reviewer and the regulatory body that, this is the data we have currently. We are considering further data, but, we are mitigating those risks, and we believe that the residual risk, is acceptable for pushing this product out and trying to seek compliance.
Okay, great comments there, Thor and Nick. Here's our next question. If a medical device has manual cleaning or automatic cleaning claimed in IFU, is it enough to prepare devices for biocomp after lifetime with manual and automatic cleaning separately, or do we have to consider sequential manual and automatic cleaning for preparation of articles for biocomp?
Yeah, this is a great question. And like I said, I don't know how this is going to be enforced, but like a lot of people who I've talked to, and there are some companies who are actually looking at already doing this for cleaning and disinfection, they'll do repeats. So they'll do their validation like it's normally done, the 6 cycles, soiling, non-soiling, and then they'll basically run just repeat washer disinfectors or whatever they do, cycles until they get to an end of life. And then they perform biocomp. But I get a lot of people who also question, could the worst case actually be in the middle, right? Like maybe you have something break down that exposes and then it gets washed away later. Like I think there's a lot of potential questions there. But right now what I see people doing is just doing repeat cycles until they get to an end of life and then perform biocompatibility evaluations. And by the way, that doesn't necessarily mean testing, like animal testing again. It could mean, once again, the chemistry stuff we talked about, just evaluating what could happen to the materials over time, look at detergent residuals, things like that.
Okay, great. Here is our next question. What is the overlap here between foreseeable misuse and contradictions? Was this a topic for discussion during the standards revision development?
Yeah. Nick, do you want to take this one in the beginning?
Yeah, so this is interesting. I think in some cases, the reason why we went this way is we know as, manufacturers that we are not ignorant to the fact that end users, practitioners are potentially overusing devices or using devices, off-label, right? So I think this is a healthy approach to trying to capture as much risk mitigation as possible without having to, kind of almost like, I think of it as like a sterilization validation, right? Without over challenging, right? Without being unrealistic and actually kind of walking into the lines of contraindications, right? And knowing that these are reasons why this device should never be used. So again, exploring what it looks like a bit off-label, knowing how a device may be misused or potential failure, again, right, failure rates, and then understanding how we are mitigating those risks without having to necessarily go too overboard or too unrealistic into stepping into the world of contraindications for the devices, if that makes sense.
And I agree. And I do think there are a lot of customers of ours that contraindicate certain things, like do not use in pregnant women, for example, because you don't want to look at reproductive toxicology, or do not use in children or whatever. I still think that has a role to try to help. I think actually what Nick says, I think it's like, hey, if you guys know that this is being used even against your contraindication, I think there is some responsibility to assess the risk to those patients. But I think this is going to be an interesting thing to see how it gets enforced.
Okay, a couple. Well, here's the next one. Now that MMP has been moved out of the testing tables, how can we convince the FDA to follow suit? Otherwise, will we still need to keep doing it?
Yeah, good question. So the FDA was on board with, so they're the ones that compromised by putting in the language where it has to be materials that are known in medical devices. Now, once again, we'll see how the FDA recognizes the standard, if it's partial, if it's full, which I think is more of a likelihood to be partial, and then what parts they recognize. But right now, I have a feeling that this would be a part that the FDA would recognize. There's some, like I said, there's some really hard to argue data about this test. So I think that it's all going to be regulatory accepted throughout the world, but we shall see.
Okay, we have time for maybe 1 or 2 questions. Here's the next one. Does the new standard address the lifetime biocompatibility of an implantable device, i.e. the question whether the implant remains biocompatible over a long period of time inside the body?
So I would argue, and I know that this is probably also a gap for some things, but I would argue that even the previous standards attempted to address the risk of a permanent implant device in the body. That's where we run things like a 13-week implant, or other, even some of the large animal functionality studies to look at those endpoints. Now, there are gaps, as the metal hip implants showed us a while ago, that there are certain, if you want to argue what is biocompatibility and what's not, but there are certain risks that we can't address necessarily through the current testing we do. But I would argue that our current standard tries to address that. There isn't any additional, except for maybe the clinical stuff, like Nick said. There is, we didn't have time to get into it, but there is some additional, guidance on how to use clinical observations to look at biocompatibility. But other than that, I don't think we've really increased that area of things. Nick, what do you think?
Yeah, and all I would add to that is kind of how you mentioned there, Thor, that there is a little bit of guidance within this update to the standard, which kind of points to the use of post-market surveillance. So as we are creating line extensions or product changes, over time, it may be hindered a bit with new product development outside of the realm of line extensions. But for devices that have post-market surveillance data that can be leveraged, right? Complaint rates, things like that, to be added, I think is another way to kind of assume how we're looking at biocompatibility of those long-term implants as well.
Okay, we have one more question that we can get to. And again, thank you for all of your questions. We will pass these questions along if we did not get to them to Thor and to Nicholas, and they will be able to address them offline as well. So our last question will be, will the appropriately qualified requirement also apply to reviewers/notified bodies reviewing the biological evaluations?
I've had this question like 5 times. So
my favorite question of the day.
No. There is, the assumption is, I'm going to be very cautious how I answer this question. The assumption is that if the individual is working for a regulatory body, that makes them qualified. So I would hope, though, that the same requirements that go into the people who are writing these documents should be applied to the people reviewing the documents too. So, I have lots of great close friends in the FDA who I think very highly of, and the notified bodies who I think very highly of. So I think they definitely have people who fit those qualifications. And so that's my answer.
Yeah, and the only thing I would add to that too is just kind of a reminder as we write these evaluations. I think there is always a really good, understanding to make sure that we are drafting these written documents in the way that we can tell a story about the device, right? We have to remember that we've been working on this device maybe 1, 2, 5, 6 years, right, internally. And so we know this device incredibly well. We have to always assume that this reviewer, this will be the first time they've ever seen this, especially a novel device or a novel material, a novel use case, right? So always remember the story we're telling as we write these evaluations and how we draft these. And in kind of an effect to make sure that we are basically spelling out for the reviewers why we are doing things, right? So, another reason why the standard shifted to this way is, here it's a good example, is systemic toxicity, right? So the new charts moving forward will no longer have individually listed acute, systemic, subacute, subchronic, etc. It'll just say systemic toxicity, and it's our job as authors to explain why we're choosing the route we're choosing. So I think the more we explain the why and the more we explain the how will also help us kind of meet the reviewer halfway in the proper understanding and evaluation of these devices.
And just to add one quick, I know we're out of time, but quick thing, you gotta remember these reviewers are reviewing multiple submissions at one time, and these submissions are huge and complicated. So like, to make it as easy as possible on them, to Nick's point, but we gotta be as clean and clear and concise as we possibly can be. It's not a time to make us sound as smart as possible. So yeah, but that's a good way to answer that.
Great. Well, thank you both, Thor, Nicholas, for that very thorough, informative presentation. There are a lot of questions still out there, and we hope to have more content as we get more information on this topic. Nelson Labs will put out more information. We'd like to thank you, our presenters, for their insights. We'd like to thank you, the viewer, for attending this session, and we hope that you found it to be a valuable experience. This will be available on demand on the Nelson Labs website. It will also be posted into the Sotera Health Academy as well. So we do encourage you to visit either location for a review of this presentation. Again, we thank you for joining and have a great day.